Thank you very, very much for the introduction, and also thank you very much for everyone. I'm delighted that this is now the 5th joint meeting on MS in what has been called now the MS Summit. And although some people are still at breakfast, I think yesterday was quite a full meeting, and it's going from strength to strength. So I'm delighted about this. I would like to thank the organizers, thank the PCO, the company, and all the attendees for making this happen. I'm delighted to be here, and we made, in discussion with Professor Hassan Hosni, we made a bit of a change to the program, focusing on biomarkers and looking into some of the lectures that were yesterday about biomarkers and that will follow today about an aspect from my specialty, from neuroradiology, and the clinical view on that. So over the next half hour, so we're talking briefly at the beginning with definitions, so to set the scene and to be clear what we're talking about, and then looking at the principles of clinical adoption. So imaging is, of course, I don't need to say this to this audience, is key for the management of patients in general. Diagnosis, some of the diseases can only be diagnosed with imaging. The staging relies on many diseases. Closer? Closer. Okay. So staging, monitoring disease, like in MS, the planning of treatment, and the assessment of treatment response all rely on imaging. But most of this is usually qualitative. We don't measure. So quantification could possibly transform how we are doing neuroradiology. Why? It is objective, it is observer independent, or this is how it should be. It would then be reproducible and could be a gold standard or a reference. So people are talking about biomarkers. What are biomarkers? What is the definition of biomarkers? Biomarkers, it's a biological measure, and I have to underline the word measure. It's a measure, it's a characteristic that is objectively measured, that is evaluated as an indicator of normal biological process, pathogenical process, or response to an intervention. So that was a biological measure, biomarker. Okay, so that was biomarker. Now, quantitative imaging is the extraction of quantifiable features from medical images for the reasons I mentioned earlier. Okay? So let's combine the two. Quantitative imaging biomarkers are therefore objectively measured characteristics derived from in vivo images and are indicators of the processes that I mentioned earlier. The word biomarkers has went through an inflation, and therefore people sometimes are not aware or don't use it in the appropriate context. And certainly in the field of radiology and neuroradiology, sometimes the word biomarker is used for any indication of a diagnosis, but not as a measure. A biomarker is a measure, otherwise it's not a biomarker. Biomarkers are also subdivided into categories. They can be used for, They can be used to look at prognosis. The laser doesn't show you, but you see it on the slide, for prognosis, diagnostic, or theranostic, which is about treatment development. So in this slide, which doesn't come out completely, they looked, it's an older paper, where the authors looked at the number of biomarkers which then went into clinical use. And it was over 500,000, and only 100 went into clinical use. And they thought about what are the reasons? Why do we have so many biomarkers publishing, talking, and looking into research, but none of them, or only a small fraction, has been used clinically? One of the reasons is that the use of the specimens are often not ideal. Whatever that is, specimen with a CSF, serum, or whatever, is often not ideal. There's a rush to get some specimen and a rush to get them analyzed. So the plea here was, we cannot advance without having a common standard and a cross-disciplinary systems-based approach. And this is a slide from UCL, from our work. And you see on the left side, I don't know if you see the arrow, yes, you see that. You see on the left side from this line, this is the UCL this is the research side. On the right side is what went into the clinic. So you see a lot of things were developed. Yes, okay, thank you. So a lot of things were developed and trialed and published, high publication impact factors, but only a very few went into local adoption and then national or clinical adoption. Only a small number. And that's the problem, and that's similar to what happened in the pharmaceutical industry. So what we need to do, we need to have rigorous evaluation of a technical level, technical performance, but also on the clinical performance. And that requires the use of a precise terminology. I will not go into this, but everyone needs to agree what means exactly precision, what means repeatability, and so on, so that when we publish and when we work together, we are clear about what everyone means. So an example is about the precision. What means precision? How much of the measured change is due to technical variability? If I repeat it on the same scanner, or repeat it on a different scanner, or in a lab, if this is lab dependent, do I always get the same? If there's a variation, what is this variation which is technically based? Because anything biological, clinical must be beyond that. Otherwise, I won't be able to measure it or to draw any conclusions of this. So this is where an algorithm was developed. So you have here the bias and the precision of the image values, which is determined by the status of the patient and the acquisition of the scanner. As an example, if I want to measure the volume of the brain of the same patient can be dependent on whether they got steroids, so the volume shrinks, or if they were well hydrated, if he was fasting, sodium, less water in the body, and so on. So this is what is thought about, this is what is the bias in terms of the patient and the acquisition. We can control for that to some degree, but not 100%. And then you have the changes related to the algorithm itself. If I use the algorithm in the same patient number of times, in different circumstances, will it give me the same? Or if it doesn't give me, what is the difference, which I need to know? So therefore, we suggested a few years ago to follow certain steps before adopting the software or hardware into the clinical field. So first, we need to establish the needs. Quite often, we have the software or hardware or program coming to you, neurologist or neuroradiologist, asking, can you make use of that? That's the wrong way around. They need to answer the question that we have. What is my problem? What is my problem? There's an answer. But give me something, then try it. This is often a waste of time. And then things must be automated. If I need to go to a workstation, I have 100 scans to report. If I need to go to a workstation for every 5th or 7th examination, I will not do it because my workload is immense. I need to have a good way of communicating the results from radiology, neurology to neurology, for instance. And then the other thing, I have to have validated this software before using it. Also locally, I wouldn't implement a new software without having tested it in my own local environment, and then I need to integrate it in the clinical workflow and make an in-use evaluation. So let's look at some of those. Establish the area of clinical need. So that means I need to identify an appropriate imaging biomarker for which is disease specific. I need to demonstrate the basis, define the purpose, and define how it influences the management. A biomarker, to use it clinically without you making use of it clinically, deciding what the influence on management is not useful. So let's look at MS. We have a number of biomarkers, very many biomarkers. They can be based on MR, PET, OCT, fluid biomarkers, oligochronal band, CSF, serum, and so on. So they represent, of course, different aspects of the disease. The advantage of MR, as you know, it's an objective measure, it's pool, it's used all the time in very different contexts, when we want to make the diagnosis of MS, follow it up or follow up the monitoring. And you all know that we have two overlapping pathogenic components. You start with an inflammatory pathology, which then moves into neurodegeneration. You have both from the beginning for a long time, but the main one is inflammation at the beginning and later on neurodegeneration. So my biomarker will assess different things at different times in different phases of the disease. And here are some examples. What can we do? We all worked on T2 mapping, T1 or T2 mapping, relaxation times, of course, myelin water imaging, DTI, diffusion magnetization transfer, but looking at the clinical maturity of these, and this was published by our group some time ago, we looked at the clinical need. Yes, all these, which I mentioned earlier, do meet certain clinical needs. They went through idea and proof of concept. Some of them went beyond reproducibility, but none of them went into defining clinical normative values cutoffs and being used clinically. So a lot of work, a lot of measures, but none of them made it into clinic. So what is the one marker that you use to define your treatment, to stop treatment, to change treatment? At the end, it is one thing, the new lesion. Over the last 20 years, it is just the number of lesions, the new lesion. This is what will make you change the treatment or stay on with the treatment. So we have the gadolinium lesions, as you know, which are indirect markers of inflammation, T2 lesions, which reflect permanent footprint. And we can see that, of course. Okay, so the lesion volume, if you think about T2 lesion numbers, the lesion volume is important because it defines the prognosis. If you have, that was paper that you will know probably, that at the beginning you have a higher lesion load when you come up with this patient comes with CIS or first diagnosis, the chances for that patient going into relapsing, remitting over a quicker time is much higher than if it is absent. So the next thing is, of course, well, let's look at lesion volume. Let's have an automated method that measures lesion volume. You look at here at 2 time points, and you see that this here is smaller after 6 months, but there's a new lesion here. So these two changes, if I do the lesion volume, almost cancel each other. So if I look at the lesion volume has decreased despite me having new lesions. And therefore, the lesion volume is problematic on its own, without the context, without seeing the numbers. So, in terms of then looking at the lesions themselves and saying, okay, the lesion volume is confusing. It's difficult to compare between scanners, between different software levels, and so on, between manufacturers. Let's look at the individual lesions. Well, we know that the inter-rate variability is not insignificant. I can train people, not only doctors, also nurses or programmers, but the consistency is difficult and is questionable. So then we are all working on getting this automatic co-registration, and this is one of the examples from Siemens, where they co-register and then color-coded and then come up with a way to visualize the results. Okay? So this is quite nice because with that number of lesions, you may miss this one to be a new lesion. However, with color coding, it's even easier. However, I find this terrible. This is not something that I find easy in my daily routine report if I get this for every patient. This must be much more simplified and much more appealing. But this is the step that we'll work on, manufacturers, centers, on getting something that identifies me, the new lesions, and therefore provides a better objective measure. So, going back to the concept, and the concept, second step was to design the algorithm, compile reference data, and this is specific to every disease. It is specific to the biomarker, and the next step then is to communicate the results. I will not go into too much detail about this. And then the next step that people tend not to do is the pre-use validation. So I need to validate things, as I said, clinically, and I need to look at credibility, yeah, and the performance. Credibility means, does it make sense what I'm seeing? Does it keep the right side? Some things are very simple. The right side, the left side, the left side, and so on. Does the whole thing make sense, for instance, in terms of the volume of the lesions? And then integrating it into packs and having it go automated and done this. So I'll give you another example on hippocampal sclerosis. You know that the diagnostic challenge with hippocampal sclerosis is to look at the signal intensity and the volume of the hippocampus. And the problem is for me, as a radiologist, neuroradiologist, that the patient sometimes like this or like that in the scanner, and what I do, I compare the left side with the right side, so I might have problems in making the right assessment, and therefore there is a clinical need for something that delivers me something automated. So this went through a process over years. First, showing that this algorithm is sensitive to pathology, then developing an algorithm that looks at both the volume of the hippocampus and the signal intensity, developing something that's automated in the quantification, validating the pipeline that was developed and established, and then presented in an appropriate way that's easy to use. And the novel thing here was to measure the hippocampus along its whole distance. So not only on the one section, but along the distance from anterior to posterior. And this is now the visualization that we did for the patient, so that you see you have the reference population here in violet and the standard deviation, and so you see whether the volume is below, and you see here whether the signal intensity is higher than normal or not. So that's easily accessible and easily seen. For all this, when you go into PACS, something very practical, but it's not straightforward. You need to provide some explanation to this when you put this into PACS or provide the patient with that kind of information. So within that framework that I mentioned earlier, we have here established preclinical need. We have developed a method for automated analysis, developed a method to communicate the results in a way that is accessible, and then looking at the technical and clinical validation would be the next steps, as well as the integration and the in-use validation. So for the pre-use clinical validation, for instance, we looked at the correct, incorrect designation. We saw that there was an improvement for everyone, which was strongest for the non-medical staff, non-doctors, then for the trainees and the least for the consultants, which makes sense because the experienced consultant will improve a little bit, whereas the trainee will improve much more. The problem is if you have a disease on both sides, but here also the accuracy improved significantly, and also the agreement improved significantly. So overall, an improvement of the clinical workup. And that's another example now from the clinic. We introduced now that this in the clinical routine, and for every patient with hippocampal sclerosis. You see here, you don't need to know much that this is too small and too bright, but this shows here in the segmentation. And then if I go to the next slide, you see here that on the right side, the volume is too small and the signal is too high, confirming the diagnosis. So now I get this automatically, see it with it, and then make the overall assessment. Same here, I don't see any difference. This was confirmed by our algorithm that it is normal, and then you have the additional confidence that it is normal. Okay. So what is left is now the in-use validation, and this is what we are currently doing, but it's not finished. And then, of course, you need to get it to other centers and have it working not only on one MR scanner, which is the next problem, but currently it's expanding it to all other scanners. And that is one of the problems that we have. If you measure the temperature, whether you measure it in Cairo or in London or whatever, it's the same, or it should be the same. So this is calibrated. With the MR scanners, it is not. It doesn't provide you with a measure that's everywhere the same. The CT does. CT gives you the house 2 units. But everyone is working now on trying to turn the MR scanner and something that's quantifiable that can be shared across sites like thermometer. So when you look then at what is made available from the industry and what has been, for instance, cleared by the FDA or is being CE marked, the focus has always been on the technical validation. There is a very limited clinical validation, and there is not at all any kind of workflow or in-use validation. So they come to you and tell you, I have a wonderful thing, and they tell you this is FDA proven, but that doesn't mean enough. It's not enough for you to buy it and use it in the patient. You need to assess it in-house and look at its clinical validation, at least in the environment. And this is one of the examples that we wanted to start with dementia and then use it for multiple sclerosis. So this is looking at the volume, and we have here a nice overview of the brain and the volume, so right side, left side, and you see the various areas very easily where the volume has decreased and where the volume is where it is. The last step, everything was done, published, the various steps that I mentioned earlier. When we came to the final step of another test, just to see it makes sense, it failed in 4 out of 20 cases, so we had to go back to the drawing board. So there's a reason why some of these software and hardware are quite expensive, because if you get to that stage, it takes a lot of work and you have a lot of failures until you get to the last point. And I give you one simple example about quantification. In a very simple manner. You all see, I think, you probably all see that there is here the tumor which shouldn't be there. So that's an acoustic neuroma in typical location. And that was the scan from 2017. This is the scan from 2020. It looks different. And someone measured and saw, okay, well, this is getting smaller. And from 14 to 11 millimeters. And in the report, focus on what is in red, they said there's a slight decrease. The maximum is 9, 10, 12 to 14. And the question is, has there been treatment? What happened? You know, tumors don't shrink. Why did it go down? However, you look at it again, and you see the same applies, yes, in the coronal view. It's getting smaller, but look at the shape of the brain. Look at the shape of the brain. The shape of the brain is completely different. You look at this here, and you look at this here. So the patient's brain, of course, hasn't changed. It's the images that provided you with a different image. And why has that happened? Well, this is one of the scanners which we are using as a mobile scanner outside of our main department, and which has its own company. And they had a problem with the quality control, so they didn't do it. And therefore, we suddenly had to go through a big process. It's in the UK, in London. On the 18th of September, the MR superintendent was informed that the mobile blah, blah, blah had a calibration problem. Okay. Radiologists had concluded that the skull dimensions of patient scans were significantly smaller. Concerns were expressed that this could lead to issues when comparing measurements. Suddenly we had a big problem. We have been scanning for 2 months in a way that didn't take account of this quality. So we had to produce a report. It's not so easy. We cannot just get away with things. So we had to produce a report of actions taken following MR calibration concerns. In this, we identified, measured the days from the 1st of August, 2nd of October. We did an audit. We found over 560 patients who were scanned. And one radiographer went to all those 500 and something reports and looked at the risk, low risk, high risk, and so on. And the high risk were those where a measurement was given, and you saw the measurement was wrong. So we had to go through those patients where there was a measurement, and the measurements were all incorrect. All those patients where there was an incorrect measurement, especially those who had a tumor, had to be recalled, and we had to rescan them and do an amendment to the report, which is why, when you get back then to 2021, suddenly the tumor is of the same size as it was in 17 and is not shrunken again. So this is why we intrinsically, all of radiology and neurology have been trained to be suspicious of measurements. This is how we are trained. We have something in our brain that our brain is able to do, putting things into context and analyzing that. So that was then in the reports, and therefore one of the things is that the quality of assessment is not as bad at all. It's actually quite strong, and it's very hard to beat by a machine, although we think that we are there on a number of fronts. And there's a saying in English, which is, show me a radiologist with a ruler, and I'll show you, a radiologist in trouble. So, and that is something, as I said, we were trained from the beginning. Just have a few minutes. So, and I show you radiologist in trouble, and that's in German, and it means the one who measures a lot of rubbish. They play on the word. So you have to be very careful about this. So when you think about full automation workflow, when you think about introducing imaging biomarkers into the clinic, you have to have full automation. You have the workflow integrated, and that's key. Because anything else is time-consuming, will not be used. It must be generalizable across MR scanners and protocols. So so the potential radical transformation of report and patient management is possible, and I think will happen. But the clinical validation is key, and full automation is key to achieve that. And with that, thank you very much.